Bismillahirrahmanirrahim. So we are coming to the estimation of hemoglobin or determination of the hemoglobin concentration. So now we know hemoglobin, heme, the iron containing heme structure, and globin, it consists of, it's a non-protein portion, and it consists of, the heme is a non-protein portion or non-protein pigment which contains a central iron atom, whereas globin, it is composed of 4 polypeptide chains, 2 alpha chains, 2 beta chains. Now, what are the main functions of hemoglobin? First of all, we have transport of oxygen in the form of oxyhemoglobin. 4 molecules of oxygen are carried by 1 molecule of hemoglobin. Then transport of carbon dioxide in the form of carboxyhemoglobin, and that is 23% of total carbon dioxide transportation. Now coming towards regulation of the blood flow. Regulation of blood flow is very much important because a nitric oxide, this compound, it actually binds to the hemoglobin, but when it is not bound to the hemoglobin, it is actually a vasodilator. It causes vasodilation, as a result of which blood flow increases. Vasodilation, it means that the narrow capillaries, the narrow ones, they elongate, they enlarge, as a result of which it controls the amount of the blood that is flowing, and it's a buffer because it transports carbon dioxide and it prevents acidity in the whole body. Now, synthesis of hemoglobin. There are various steps in synthesis of hemoglobin. So this diagram is good for the steps. 2,6-sinyl coenzyme A plus 2 glycine, they form a pyrrole, and 4 pyrrole ring make protoporphyrin 9. Then protoporphyrin 9 plus iron makes heme. Heme plus polypeptide, they make hemoglobin chain, that is alpha or beta, and 2 alpha, 2 beta, they make hemoglobin A. Hemoglobin A is the most abundant hemoglobin in humans. Now, the molecular weight is 64,440. 34% wet weight, 95% dry weight. Also, hemoglobin can carry oxygen since it contains 2 alpha chains, 2 beta chains. One chain will bind with one oxygen, so it will form it can carry 4 oxygen molecules in their molecular form rather than their ionic form, and 1 gram of hemoglobin can combine with 1.34 milliliters of oxygen. Now let us determine some adult and fetal hemoglobins. Adult hemoglobin, the major one is hemoglobin A, consisting of 2 alpha, 2 beta chains, whereas the minor one is hemoglobin A2, which consists of 2 alpha, 2 delta chains. Fetal hemoglobin, it has 2 alpha and 2 gamma chains, whereas the intraembryonic, intrauterine one is the Gover 1 hemoglobin. It consists of 2 zeta and 2 epsilon chains, whereas Gover 2, it consists of 2 alpha and 2 epsilon chains. Gover 1, the first and foremost one. After that, Gover 2, in which zeta will be replaced by alpha. Then fetal hemoglobin, where this epsilon will be replaced by gamma. Then the original one, where alpha and beta chains will appear. And the minor one, which has 2 alpha and 2 delta chains. Now, derivatives of hemoglobin. We have the oxyhemoglobin, hemoglobin bind with oxygen. Now, methemoglobin, hemoglobin plus oxygen plus potassium ferricyanide. Carbaminohemoglobin, in the carbon dioxide plus hemoglobin is carbaminohemoglobin. And self hemoglobin, the name suggests it is going to be with hydrogen sulfide H2S. Now, estimation methods. We have two methods. One is the Sahli's method, other method is the spectrophotometric method. Let us consider the Sahli's method first. So, as you know, firstly we are going to draw blood. We will have a clear swab through which we will prick the blood via lancet, then use N/10 hydrochloric acid to convert the hemoglobin of the blood into hemetin, as a result of which, also when this hemoglobin will be converted into hemetin, and it will produce a golden brown color, and this color depends on the amount of hemetin produced, which is directly proportional to the amount of hemoglobin produced, and hence it helps us to actually identify the amount of hemoglobin that is present. Let's go to advantages and disadvantages first, then I will show you its whole apparatus. This Sahli's method, it's simple, it's less costly, it is minimal, and it can do massive surveys. But for disadvantages, First of all, the hemetin is not in the true solution. Secondly, it only determines oxyhemoglobin, but other forms, deoxy, methylmohemoglobin, they are not measured through this. And degree of error may be high if proper precautions are not taken. They are saying that if there's human error, there's a chance of this. In this method, there's a high chance of human error. For spectrophotometric method, you need to know the names only. They are oxyhemoglobin method, the halidane carboxyhemoglobin method, Wu alkaline hemetin method, and cyan methylmoglobin method. So coming towards the Sahli's hemoglobinometer, we have the comparator box. I will show you in a moment. Then hemoglobin tube. There will be a red color bead in it. Then hemoglobin pipette. Then a stirrer, a pasture pipette, and distilled water. Also N by 10 in HCl will be required. And there will be a graduated hemoglobin tube. Now, firstly, you can see a glass rod or a stirrer. Then we have this hemoglobinometer pipette. Then we have this graduated hemoglobin tube. Then we have this comparator box. This comparator box has this hemoglobin tube fitted into it, and this graduated hemoglobinometer tube will actually help us identify the amount of hemetin which is produced, and this hemetin will help us in calculation of the hemoglobin concentration. You can clearly see over here, this is the hemoglobinometer tube, the comparator box in which the graduated tube will be placed. Then this is the stirrer, and this is certain type of pipette through which you can add the blood into it. Now the amount of hemoglobin which is present normally in males, it is quite large amount, that is 13 to 16 grams. In females it is 12 to 15, one number less than the males, and in children it is greater than males even, it's 14 to 19 grams per deciliter. Why it's greater? Let's find out. So the first question we have is why hemoglobin is lower in females as compared to males. Firstly, because of menstrual loss, that is 20 to 30 milliliters per month. Secondly, we have the estrogen, which has an inhibitory effect on erythropoietin, which actually stimulates red blood production. So it has an inhibitory effect on this much. Why the males have more? Because androgen, mainly the testosterone, they have a stimulatory effect on erythropoietin. Why is hemoglobin high in the newborn, which is 20 to 22 grams per deciliter, or 14 to 19 grams? The main reason for this is they are living in a state of hypoxia. This state of hypoxia causes stimulation of the erythropoietin, due to which hemoglobin levels increase. But when a child is born, the hemoglobin levels start declining until the adult levels are reached. Causes of increased hemoglobin level. Physiological causes, they are males, newborns, and high altitudes. And high altitude also causes hypoxia. Newborns also have hypoxia. So pathological causes, they are hypoxia and polycythemia. They can be due to heart failure or certain lung diseases. Now coming towards decreased hemoglobin level, they are due to pregnancy. In pregnancy, in the previous the packed cell volume experiment, we also saw that in pregnancy, due to hemodilution, packed cell volume decreases. Also, hemoglobin level also decreases. Pathological causes are all sorts of anemia, which actually reduce the amount of hemoglobin in your blood. Now, this is also the Sahli's apparatus that's mentioned in your book, the comparator box, where the graduated cylinder will be placed, the graduated tube. Then this was the stirrer. Then we had the hemoglobin tube, a pipette, and then this is the N by 10 HCl. So this is how you can learn the Sahli's method for the determination of hemoglobin. I hope you understood all of that, and if you learned something new, please subscribe to my channel. Thank you very much, and Allah Hafiz.